postdoctoral training as a research fellow at the Center for Neurological Disease, Brigham and Women's Hospital, Harvard Medical School, Boston. Dr. Awasthi has held academic appointments at prestigious institutions, including Harvard Medical School, contributing to fundamental advances in T cell biology in autoimmune and infectious diseases. With a citation index exceeding 15,000 and around 20 plus publications in top tier journals such as Nature, Science Advances, and Lancet Infectious Disease, his contributions span fundamental discoveries in Th9 and Th17 cell subsets, anti-tumor immune responses, and vaccine development. Dr. Avasthi is honored with the G. N. Ramchandran National Bioscience Award, the CDRI Award for Drug Discovery, and the Young Scientist Platinum Jubilee Award. He also served as the Secretary General of the Indian Immunology Society and Executive Council member of the Federation of Immunological Societies of Asia Oceania. Today, Dr. Avasthi will be enlightening us with the clinical trials and future trends in epigenetic therapy. On behalf of the Department of Biotechnology, Guru Hassan Vishwavidyalaya, I request Dr. Santosh Kumar to welcome him with a book. On behalf of the organizing committee and all participants, please join in extending a warm welcome as Dr. Amit Awasthi takes the podium for today's lecture, sharing his expertise drawn from his groundbreaking research and his informative scientific views.
Thank you very much for your kind introduction. You guys are comfortable English, Hindi both, or okay, great. And I take this opportunity to say thanks to Santosh, who happened to be my friend now, but he was a junior when I was doing PhD from the same place in NCCS Pune, and subsequently he went for postdoc. I went to other place, and then again we got reconnected for some good cause, trying to do something which can help Indian patient in a way that we can treat people in a way for cancer and maybe other conditions. So I will be trying to be as basic when I talk about the T cells. My lab is primarily working on the T cell research, T cell biology, and T cell, as you know, are involved in many conditions, including how many of you know that T cells are the one which gives you a long lasting memory response in the vaccines. And we all know that during COVID-19 the how vaccine is so important, and when you get vaccinated you are protected for the life because you have these T cells which recognize the antigens which is on the viruses and viral antigen, let's say the bacterial antigens, uh, and these T cells is having a very specific receptors, and these receptor will recognize this small peptide. You know, these are the proteins. T cell recognize protein antigen, and these proteins are chopped into small portion by the antigen presenting cells. Uh, Antigen presenting cells, you may be knowing. How many of you know what are the antigen presenting cells? Yeah, so majority of the people know what are the antigen presenting cells. These are the cells which is myeloid origin, that contains macrophages, dendritic cells, and these cells actually take up the antigen through the process of phagocytosis, right? Once the cells actually engulf the bacteria, then these bacteria goes to phagolysosome, right? And in the lysosome there is acidic environment, and in the acidic environment there are enzymes which are activated in the acidic environment, and they cleave this bacterial protein into small portion of the peptides, and these peptides now bind to MHC molecule. How many of you know MHC molecule? These are the major histocompatibility complex, and these MHC molecule within this ER, and they actually bind to these peptide through a affinity, Affinity means that you have certain amino acid on the MHC molecule, and you have the peptide which is having certain amino acid, and they bind to each other because of this molecular interaction. And because of this affinity, these molecule bind to MHC, and they travel from the cells to the cell surface. When they go to the cell surface, if let's say if you have a MHC molecule which is not able to bind a peptide in a kind of a manner, optimal affinity, what it will happen, the MHC molecule will remove this peptide, and it will try to find the right affinity peptide. So this kind of multiple permutation combinations are happening within the cell, within the antigen presenting cells, which are macrophage dendritic cells. And once you have the proper affinity peptide which fits with the groove of MSC molecule, it gives a signal to take this peptide, or antigen rather we should say, to the surface of the cells. And when actually it shows this on the surface, then you have T cell in your secondary lymphoid organ, right? So the T cells which are in